Up next, we've got Recce Pharmaceuticals. They're listed under the ticker RCE on the ASX. They're developing a new class of synthetic anti-infectives to combat the global threat of antibiotic-resistant superbugs and viruses, which we are unfortunately too familiar with in this day and age. We have CEO and MD James Graham presenting today. James himself started as a shareholder in Recce Pharmaceuticals, and he's had extensive experience commercializing early stage technologies. He's also assembled a team around him with great scientific expertise to create value for shareholders. James is going to take us through the presentation, I believe, today, as well as a couple of questions at the end. So over to you, James.
Yes, hello, Tim. Good to be with you. And thank you, Mark, on that presentation. I certainly enjoy bringing myself up to speed on the progress of antisense and the developments in similarly that major unmet medical needs space. So today I bring to you Recce Pharmaceuticals. Recce Pharmaceuticals is listed under ticker RCE on the Australian Stock Exchange, with a dual listing on the Frankfurt Stock Exchange or exchanges under ticker code R9Q. As Tim well says, I'm not the inventor of the technology, I'm the investor, and the investor from the perspective of backing good people and great science. The science is invented by a former head of research of Johnson & Johnson Australasia of a decade. He was in fact executive director of the board. And my initial investment was for the purposes of giving this retired gentleman's new technology out of his personal laboratory a fair go. Now, with that, we built out a team, and I have the joy of working with wonderful individuals such as Dr. John Prendergast, represented on the left of screen here, who's present chairman, non-executive chairman of a New York Stock Exchange biotech company, and lead director of a NASDAQ biotech company. And he's in fact our executive chairman with his doctorate in the study of bacteria and blood diseases. I mentioned I began as a passive investor supporting a good technology, getting a fair go. Over the time of participating or supporting that, I became more interested in fact, built out a team around, formally joined the company and board listed on the ASX, where I was executive director for the first 5 years and transitioned into now managing director and the second largest external shareholder to the founder itself. Michelle Delizia is a fantastic example of the caliber of people within our team, co-inventor of Recce Antibiotics, and we've got a wonderful suite of supporting board members, former global head of research of Johnson & Johnson, CEO of Janssen, the quality specialist of Pfizer, and so on. So what is Recce built upon? Recce is an anti-infective technology focused upon bacterial and viral diseases. We have strong intellectual property backed by patents granted all over the world in major pharmaceutical markets, as well as with a particular one of those, the addition of a FDA-awarded QIDP designation for sepsis, septicemia, or in technical terms, bacteremia, which basically means blood poisoning, and that gives an extra 10 years of market exclusivity independent of patents backed by the government in the USA. Our compound as a technology is able to be administered via various methods of administration for various indications. It's most importantly demonstrated an important ability to work and keep on working with repeated use, a fundamental challenge of all existing antibiotics to date. This is a snapshot really of our pipeline, and as you can see, there's a lot going on. And there's a lot going on because there's an infectious disease company in a global landscape of an infectious disease crisis. We have multiple unmet medical needs that we're targeting. We've broken it down to methods of administration. So in simple forms, intravenous administration here, we have clearly made the discovery. It has been validated by a suite of preclinical activities and has entered phase 1 and phase 2 human clinical dosing. We talk a little more about those further in the presentation, but it's effectively a split between sepsis, the #1 most expensive condition treated in health, and urinary tract infections being 25% of the underlying septic infection cause. Topical infections, where we have burn wound study results, and we have transitioned or expanded that topical application to include diabetic foot ulcer infections, and then a suite of preclinical compounds focused on major unmet medical needs and other areas of indication as part of a portfolio approach that this company takes. So looking to our first indication and primary focus, if I say it, it's sepsis, septicemia or blood poisoning. It's actually the #1 most expensive condition treated in health, double the second most expensive, because there's no drug therapies specifically approved for that bacterial infection, because it's unknown at patient presentation just what that bacterial infection is. That results in a 1 in 5 death rate in the community, and I've been a sepsis patient myself, or a 1 in 3 in hospitals. Thankfully, the drugs worked for me. Too often, they simply don't. And the pharma economic burden and, of course, the mortality is quite significant as a result. The reason for that is every hour left untreated, the likelihood of patient survival decreases by 8% each hour. So in my case, as with any other septic case, the patient presentation to the clinician, the first thing they do is they do a blood draw, they rush it off to the laboratory, and they cultivate it out, trying to work out what type of bacteria is in your blood and what type of antibiotic, hopefully, is that bacteria susceptible to. Until they know, which can take 24, 36 hours, it's clinical guesswork. And even if they do know what type of bacteria, there's no reassurance that antibiotics will work. So they don't know at patient presentation what type of bacteria, and there is no existing drug therapy that's able to tackle any bacteria in the blood. This is position 2 and has demonstrated ability to bind to the bacteria in the blood and interact with that, whether it be a gram-positive, a gram-negative, or a hypercellular mutated form, which gives the potential to at patient presentation, stop infection in its tracks, leading to obviously better patient outcomes, a significant reduction to that pharma economic burden, and a treatment paradigm as a whole. So this just talks about, in that visual on the right-hand side of screen, the underlying precursors to sepsis. It can be, a nail infection it can start in. It can be a stubbing your toe. It can be a knee surgery or simmer. You get a break in the skin, bacteria has every chance of getting in, and it spreads from there. So Dr. Melrose, in his wisdom, really looked to the world of antibiotics and said, all existing antibiotics are cultivated from nature. If this, then that, and if the bacteria mutates, that lock and key as traditional antibiotics are, no longer works. It's just a simple fact of nature. So beginning with first principles in mind, we said, let's do away with natural processes and let's create a compound that has a universal mechanism of action, a broad spectrum capability, and is able to work and keep on working with repeated use, something antibiotics can no longer do. What that basically means, and has been demonstrated through our ability to work against the full escape pathogen suite of deadly bacteria, is an attraction to bacterial species, a binding and interaction with them with great specificity, meaning without harming the healthy human cells. So that's lending itself to be, as we are, the world's most advanced sepsis drug candidate as a new class of antibiotic therapy. And we are naturally transitioning through that human clinical trial process headed towards market. This is where we get a little technical, and of course we can take as deep a dive as we wish here, but it's really a visual or a pictorial derived from the mechanism of action experts out of Linnaeus Biosciences. It's envisaging or representing how our compound works. In essence, we're a small molecule, about 750 Daltons in size. We permeabilize the bacterial cell membrane, and we do a number of things from there. So firstly, we shut down irreversibly the ATP synthesis, or the beating heart of those bacterial cells. Then, and relatedly, we remove the Z-ring complex, and that Z-ring complex is basically how cells divide and multiply. Well, multiplying cells is infection spreading. And if you're stopping in its tracks faster than any antibiotic has before, and irreversibly unlike any other antibiotic, and the ability for the cells to divide and multiply, you're kind of working as an antibiotic should. So that's an example of course, how our compound's mechanism of action is taking place according to those experts. And that's looking at their library of over 100 known mechanisms and saying, in fact, it was kind of, they said, we don't know how you work, but you work a bit like this, a bit like that, a bit like this, all in combination. So that was pleasing, but it's easier sometimes to just say you're in the same bucket as the others. But frankly, when the others don't work, who would want to be in there too? So this is it in literature form. This I like, if I say it particularly, because you can see at a visual the bacteria being those small little molecules there, which is the offending bacteria, and the healthy eukaryotic cells. On the left-hand side of screen, you see them without treatment, yet on the right-hand side of screen, most importantly, those bacteria on the same dish with the same exposure are not growing. In fact, not only are they not multiplying, if you focus on anyone particularly, sometimes you'll see them disappear. Nevertheless, the healthy eukaryotic cells continue to grow and multiply uninhibited, as any healthy cell should. And this is, of course, against a gram-positive bacteria or gram-negative bacteria. And if we look across to staph bacteria as the complete opposite, we see that taking place again. So that specificity to be able to discern between deadly bacteria, because there's no such thing as good bacteria in your blood, and the healthy human eukaryotic cells lends itself to a compound that's able to be efficacious and safe and tolerated, as we've demonstrated in our phase 1 human clinical studies. So I quite like that. This is just an example from our granted patents around the world of our ability to work and keep on working with repeated use. This study is against amoxicillin, which has about $10 billion in revenue each year. After 2 cycles against E. coli, a very common bacteria, that they no longer work. We took ours out to a minimum of 25 repeat cycles. This was actually one of the first studies we did. I personally funded this. So when after 25 cycles and amoxicillin and others could no longer compete, we went, oh, case proven. We've since done it many hundreds of times, and we've never found any resistance to our compound, which lends itself to our unique mechanism of action. This is our phase 1 intravascular study, which is being intravenously infusing by way of into the vascular system, starting at 50 milligrams and escalating through different cohorts of healthy males. And that's particularly looking to determine, is this compound safe and is it well tolerated? Well, we actually got up to 6,000 milligrams and beyond. I can't say we're beyond, but beyond, which is 120 times the initial dose. And it continues to show the compound to be safe and well tolerated. Saying more simply, in 1 hour of IV infusion, as this demonstrates, the human is able to receive 6 grams. That's a lot, 6 grams of product. So we're very pleased by those results to date. Most importantly, that dosing parameter is well within the efficacy range indicated in our preclinical studies. We've a suite of preclinical studies, as one could see in our previous investor presentations, demonstrating capability against all sorts of different type of bacterial infections. So what you're really looking for is a therapeutic window. How much can the patient take and where does efficacy take place? And any activity within that range is consistent with an ideal pharmaceutical drug. So this is looking to some of the PK/PD data, and particularly we're looking to concentration correlation between the compound itself and that in human plasma and human urine. And human urine, despite the terminology there, is important because it demonstrates the compound is being excreted through the kidney. So IV blood filters the kidney. From the kidney, it goes down the urinary tract to then reside in the bladder. Talking about human plasma here, clearly and importantly, we see dose-dependent increases up to a nice concentration across the 60 or so healthy volunteers. So that's good to see and in line with expectations. More interestingly, if I say it from an efficacy perspective, the concentration availability of our compounds identified in the human bladder is high. In fact, it's some 15 times higher than we would have anticipated. And consistent with that, and giving no surprise to the outstanding urinary tract or UTI preclinical studies we have achieved, this high concentration of compound in the area of urinary tract infection is an ideal location. It demonstrates the ability for our compound to get there and get there in a higher degree of concentration, which would lend itself to the probability of an efficacious outcome, which we discuss further in as a next destination. In fact, here I think we touch upon why UTIs fits in our infectious disease portfolio and model for sepsis. And that is urinary tract infections, firstly, have a high degree of resistance to existing antibiotics. 76% of E. coli infections resistant to amoxicillin. Tetracycline, which was a fairly recent antibiotic, by memory, around 71% of infections resistance to. And 25% of all UTI infections go on to cause sepsis, or sepsis is a result of 25% underlying infections. So what that really means is, starting in the bladder, as you move up the urinary tract, you're starting to obviously see infection spreading. And I'm just going through the paradigm of UTIs leading to sepsis. Kidney infections is an area that we may further move into, because that is normally where that UTI would spread to, which at any moment can break out into the blood system as a result. So having a full paradigm of patient treatment opportunity that this may represent, from the precursor UTI potentially through to the kidney infection, and heaven forbid, all-out sepsis, positions our compound with quite a nice market vertical, where the patients are in hospital. You can tackle that at early patient presentation, or have confidence that regardless of how mature their infection may be, the potential to treat those patients may come true there. So that's looking good. Stepping across to our topical side of things, this is looking to utilizing, instead of in your blood, on infected burn wound skin. This is a study that has been taking place out of Perth of Western Australia. It's got the usual burn wound experts, Professor Fiona Wood, Dr. Ed Rabi. Fiona's by example, or Professor Wood more formally, known for her spray-on skin. And this study, sponsored by the West Australian Health Department, and forgive the gory images here, has been utilising our compound sprayed upon infected burn wounds. In fact, the pathogens in these wounds have been most of, if not all of, the escape suite of pathogens. Some of them have been extremely drug resistant. In fact, one of the patients, I think, had his infection for 17 years. I can't imagine that, but that's that. And what it's really demonstrating is you can see just with these preliminary photos, the infection is moving from an infected, raw, hostile state to a stable, good skin coloration returning, which is vascular flow and recovery of healthy skin cells, removal of the crust of the wound itself, which is an ideal for wound healing, and an indication of good tissue penetration, getting deep within below the skin surface to hit underlying infection. We've presented our patents in a slightly different format here, just to really take a bit of a technical deep dive and capture of how much intellectual property is behind this. And whilst we see filing dates, it's most importantly of those granted dates, the 20 years that this intellectual property allows the company. So we have granted patents out to about 2035, 2037, in fact. Composition of matter, method of administration, broad spectrum infections of both bacterial cells and viral cells, so on and so forth. So a very, very nice suite of intellectual property there. We manufacture our compound ourselves to both volume and quality for all of our clinical trial phases. So we're well established there. It's extremely inexpensive to make. We're currently producing at about 500 doses per hour without any expensive waste or materials. So the preliminary economics associated with the underlying cost base and market opportunities that may present is very much in hand. The capital structure here is really representing, obviously, the price per share to a capitalisation of around 116 million Australian dollars. Cash and deposits, let's not be overly concerned by the 5.7 million suggested here. As we have in the green asterisks there, we're expecting an imminent R&D rebate from the government, non-dilutionary cash inflow of around $3.5 million, as well as a return of capital from other non-dilutionary sources within the existing quarter. So we really see our true cash position as around 10 million Australian dollars. We're obviously, and without any debt and caveat free, and significant capital has been attributed to bringing on those new clinical studies, which we might even touch on in this next slide. Here it is, which are UTI single patient dosing. The patients are expected first dose Q1 of next year, just around the corner. The phase 1 IV PK/PDs in healthy human subjects has allowed us to build into multiple patient dosing for a 1B/2A study in sepsis, so UTIs and sepsis, same method of administration, different indications. We then, with that, anticipate moving quickly into urosepsis. So instead of a singular administration for a more simple UTI infection, looking at multiple administrations to tackle that urosepsis or that advanced UTI stage, which is becoming septic or is in early stages of sepsis. As well as, don't know if we say it here, but not say it, we should suggest otherwise, a New South Wales leading teaching hospital on the unmet medical need and major patient population of diabetic foot ulcers. So where that finds us is we're a new class of anti-infectives with a significant intellectual capital backing. We have major unmet medical needs by way of large patient populations not currently addressed by existing antibiotics. We have a suite of preclinical data coming through to likely become clinical assets in the near term. We make our compound seamlessly with great manufacturing capability and scalability, and a large number of clinical programs actively taking place as we speak, with news flow events that would follow. So in summary, that is Recce Pharmaceuticals, and I would welcome any questions.
Thanks for that, James. No, really appreciate you making the time to walk us through the presentation. One of the questions that came in through email prior to this presentation was, what's your ongoing burn rate? I think someone, previously observed the cash balance being at about, sort of 5+ mil, but thank you for clarifying that, we perceive it to be more realistically around 10 within the next 2 months. But yeah, what's the ongoing burn rate with the number of trials that you have going on, and obviously a few partnerships? How's that looking?
So our all of our studies uniquely are R&D rebate supported. So most companies, in fact, pretty well all companies, have only activities in Australia supported by the 43.5% R&D rebate. Uniquely for us, we have what's called an advanced finding, which means all of our activities overseas that are R&D applicable are captured under that cost structure too. Our burn rate, as a monthly burn rate, our fixed costs are about $150,000 per month fixed. It's our variable costs that are well budgeted for, and it's always a classic snapshot of time zones. We commit upfront quite a significant capital outflow to a new clinical trial. Right at the end of the quarter, that was announced, with anticipation of inflow from the R&D rebate coming to materially offset that within the present quarter. So our true cash position, as we see it stated there and through the ASX, is around $10 million of cash runway. And realistically, these studies aren't overly expensive. That phase 1A sepsis study we did, phase 1A safety study designated for sepsis, that was a couple of $1 million. The phase 1B/2A, that'll be another few $1 million at a fixed monthly burn rate surrounding that of $150K a month. It's not overly expensive, with material upside opportunities made available through investing across the multiple programs as we do.
Yep, beauty. And we had a question come through from Stephen here. Is Recce 327 available in the special access scheme?
It is, yeah. So the special access scheme category B from memory, has been utilized in a number of methods of administration. I particularly allude to sinusitis, which I think in our last ASX update we touched upon, and actually Professor Phil Sutton talked about some of the animal studies in sinusitis, again showing fantastic ability to work against multiple pathogens. That lends itself to having previously witnessed, as we term, patient X, who had drug-resistant Pseudomonas infection in the upper sinuses, who had a complete patient response, and has in fact had no return of that drug-resistant pathogen, which he'd suffered for approximately 20 years, and was made available to him under the Special Access Scheme. For patients such as septic patients under the Special Access Scheme, just due to the time constraints of by the time they work out that that patient isn't responding to existing antibiotics, completing the paperwork and actually accessing the compound, can be quite prohibitive, but along the way, I think we have certain strategies to make that easier on the clinician and patients.
Yeah, and if anyone's sort of interested to read that article about basically the first patient ever treated with Recce 327, I'll chuck it in the chat now.
It's the first publicly said anyway.
Yeah.
I'm a fisherman. I cut myself all the time. I've had an infection or two.
Yeah, look, I think there's no question about the total addressable market for sepsis. So when are we expecting results from that phase 2 UTI clinical trial to be released?
We would expect the UTI study itself, which is a phase 2 study, to have the first patient dosed at the very start of next year with results. In fact, that whole study we'd expect to complete within 6 months, but certainly without being too particular to the month, we are anticipating within the first half of next year.
Yeah, excellent.
I'd expect from an efficacy perspective, the near-term clinical efficacy catalyst, unless something pops up on the burn side of things, would probably be diabetic foot ulcer infections. That's very active at this moment.
Mm-hmm, mm-hmm. And what can we sort of expect a press release about that?
Well, Christmas may come early, so we'll see.
All right, excellent. And James, sort of more on the corporate side of things, how are you actively, I guess, number one, promoting the company, obviously in webinars like today, speaking to investors, but in terms of, any partnerships that are in the works, how much can you speak to those?
Yeah, look, I saw particularly in Mark's presentation reference to Murdoch Children's Research Institute. They're a fantastic institute with a depth of capability and certainly knowledge across these programs. I see that as an example, and we've worked with them for a number of years, and is likely an area of deeper collaboration, for simple terminology, along the way. Where as time goes on and our compound continues to move strength to strength, it's been less of a push and more of a pull taking place in the context of, key opinion leaders from around the world contacting us with opportunities that the ability to work together may take place and similar. So not only does it turn into sort of almost tailwinds for partnerships and collaborative opportunities, but certainly the recognition of the challenge that antibiotic resistance and infectious diseases represents is going very well for us. Yeah.
Well, look, the team at Spark Plus, we've been following Recce Pharmaceuticals for a couple of years now, and we are genuinely very excited for the next phase of development and some of the results that are coming up in the near term. So we'll be keeping our eyes peeled. Thanks, James, for making the time to speak to us and answer some of our questions. As usual, if anyone would like to get in touch with James to, follow up, have a one-on-one over Zoom or maybe over a drink, feel free to get in touch with us and we'll be happy to make that happen.